Okay, let's continue on the issue of predicting protein-protein interaction sites. Just for recap, for reminding you, for us, what we're talking about when we say physical protein-protein interactions is really two parts that touch, the green and the antibody and the GP120 here. Because at some time point you find all of these CD4, GP120, these two antibodies in one big complex, most people who talk about physical interactions, they talk about CD4 binding antibody 2, simply because they're all part of one big complex. Here is really touching. And what we're trying to do is to predict in the green protein, the GP120, glycoprotein 120 in HIV, what are the residues that bind another protein without knowing the binding partner? So we don't know the CD4, we don't know the antibody, and we only want to say GP120 will bind transiently, so it will bind a protein that it will also let go, here and here. Okay? And the device that I showed you is essentially a machine learning device. This particular case was a neural network. For the DNA, we did it with an SVM and then showed that the SVM actually works better. So with the SVM in principle, you're on the right track for this story that you did. But sometimes it may be better to start with a neural network because it's easier to find a solution. And when you find the solution, you have a better idea what the problem is. The problem of the SVM can be, and again, remember, one of the major issues that differentiates the SVM from a neural network is the SVM distinguishes between data points that are classified oppositely, so binding, not binding, and are most similar, nearest in space. So if these points are incorrectly labeled, then the device will never learn that. The SVM will have a hard time. There's a margin in the SVM that essentially an error margin that allows you to sort of exclude some of these. But if the error is too high, the SVM has just a tough time. Okay, it may be that the problem is too hard, but it may also be that the data is not clean enough. With a neural network, you can easily find out. By the way, how could you, with a neural network device, for instance, how could you find out whether there is a sort of mistake of this type in the data set? So now we have an input vector, and the input vector, the I1, goes to binding, and I2 goes to not binding. If I1 and I2 are identical, that's easy to find out. But actually, typically we don't do this. Because you put your data, you put your features, by that you construct a vector, but then you do not look at the overlap between all the vectors. So the simplest thing you could do is to pre-filter, to look at all the ones where you have identical vectors that are classified differently. And the first step, in fact, should be, if you had these cases, throw them out from the SVM. That's trivial. Now, less trivial is a case where there's some measure of distance of the two vectors, that could be Euclidean distance or something like that, where I1 and I2 are not identical, but they're somehow similar. And now we get into the subtle thing. We cannot define, we cannot say that if it is larger than some threshold theta, then I will call them incorrect experimental assignments. Because it is not really the overall similarity of the vector. It could be that somewhere there's the proliner breaks the helix. It could be that the information is somewhere, and that by doing the Euclidean average here over the I1, let's call him k, minus I2k squared, this is the Euclidean average, and that just looks at the entire vector and may just say the entire vector is similar, but yet there is unit 42, and at unit 42 something happens, right? This one doesn't know that. That is exactly what you need the machine learning device for, and that's exactly why the introduction of such a theta, such a threshold, is impossible. It is very easy when they're identical, but those are the exceptional cases. For the SVM, the killer, the tripper, is when you have I1 and I2 that are very similar and classified differently. And similar where you don't have the unit 42 that distinguishes them. So back to the question that I had. If you had a neural network, how could you find cases where I1 and I2 are similar? What could you do? in the language of, for instance, a neural network? Anybody, any idea? Well, ultimately the question really is, can you learn it? Can you train it? So, let me go back to, this is time, this is accuracy or success or whatever you call it, and I have two curves. They signify two different data sets. Anybody stepping forward to guess what I could mean by this? It's trivial. Just do me the favor and say it.
Data set, the red curve is the performance on the training data set, and the other curve on the testing data set.
Exactly, and that is typically when the testing has plateaued here, we say this is the point of overfitting, overtraining. But does that curve give you an idea what you could do? Well, you could just blow up your neural network. Blow up means you add a lot of hidden units. And train. Forget about testing. Train and see whether you can learn it. If with a huge neural network you cannot learn it, then at least by the input vector that you have, these two cannot be distinguished. Okay? And the neural network simply can tell you, I will not get to 100% even for two things that are similar. So there you essentially use the machine learning, misuse or use or whatever you call that, you use the machine learning device to establish a proximity of two vectors. And we have done that, in fact, for this particular problem, we have first used this idea, where we in fact excluded some of the data. We could never train them in a huge, we blew up a very big neural network, so blowing up means a lot of hidden units. Or plus, what in your example is the 512. Whatever gave you the idea that 512 is the maximum number of units you can have. I'm sure I could come up with more than that. But so you blow up this input space, you blow up the hidden space, and whatever you cannot train then, if you run with a neural network, and with a neural network typically this is not for running for a week. This is typically much shorter. Well, with your 500 proteins, 353 plus similar match, how many resis you do have something like in the ballpark of over 100,000, right? 200,000, but the two together, plus the negative set may go up to 200,000 or something like that. But still, this is still somehow manageable. And again, so what we essentially did by that is exclude things that didn't work, and then we jumped at the SVM with that. This is one way in which you could use the device. Again, back to this issue, just to repeat, every of these blurbs here is 21 units, so the 20, and that's most likely similar to what you use for the DNA prediction. So 20 for the 20 amino acids plus 1 for a spacer. Everybody understands the spacer? Yes. And then we replace that by the profile. We add in secondary structure prediction, self-indexability prediction, and so forth. We get coverage, correctly predicted versus observed, correctly predicted versus predicted. I refer to as accuracy. You had the recall precision here for this one. This is not for the positives. Here is for all. This is your last column, last row. Sorry. Sequence only in light blue. Everything together, sequence profile, and 1D prediction here, random here. Again, there are different ways of playing random, and they actually give different results. When we looked at what makes things so strong, we found that this was largely the hotspots. And again, to go back, this is how much what contributes, and you see that evolution alone is the strongest signal. The strongest win. Everything together is the best. Now, here we get to the next question. Now we have a method that takes every single I'm sorry. Takes every single protein, can take every single protein in a genome, in human for instance, and can predict the number of binding residues. Okay, what could we use that for? Somebody who wants to work on a particular protein, we can predict it for that person. An experimentalist can design the way they do the alanine scanning to knock out experiments, and that's what people used it for. But what could I use it for on the level of the entire human genome? I could predict all the binding sites. What could it buy me? So, our idea was, let's just see whether we can get hubs from this. And for this idea of hubs, let's just get another detour. The network distribution of protein-protein interactions is the story here. The question is clear. Will all proteins, human has about 20,000 proteins, will every single protein have a similar number of interaction partners? Yes or no? So, you know biology, somewhere in the gray, the answer will be in the gray, a little bit yes and a little bit no, but I want you to fall on one of the two extremes here and argue it for you. Who jumps?
The question is like, yes,
that is the question.
Some have more or some have less. And
that is the question.
Number. No.
No, why not?
Because we really have several proteins that are actually very important, like ribosome, for example.
And that's an interesting concept. So how do you define the importance of a protein? One way could be by the number of proteins it interacts with. Can you think, since the word came up, important, what else could you use to define important protein?
Use pathways and see whether the protein has a major role and how many, like...
So that could be like you have a knockout of this particular protein, and that will lead to a damage because it has some central position or central role in a pathway. That's another definition. What else could you think about? Which could be related to all of these, but may not be. Well, one way of looking at it is to say evolutionary conserved, so something that has been around since life exists. So often people refer to these as housekeeping proteins, or housekeeping genes, housekeeping enzymes. So the kind of things that do processes that every single organism has to do. We have to synthesize amino acids, or there are certain ATP, so there are certain things that are sort of defining, in some sense, defining life, and you need proteins for, in fact, making those happen. Could call those the most important proteins, because without that, you could never construct the simplest life form. So there are different ways of defining it. Okay, but the answer now stands in the room, no. Proteins, for instance, important proteins, or some proteins will have more interaction partners than others. Is everybody agreeing to that? Okay, if everybody is agreeing to that, then that is clear. This cannot be the answer, right? Now, will the answer that? Will the answer with that, or will the answer be something else? So the green and the red and some other curves you could imagine are compatible with a no. Or you all said no, I give you at least two curves for that. What do you say to these two curves? And let me bring up another question that is slightly different. So I suggest you plot the size of settlements. So settlements, towns, cities, megacities in the world. How would that look? The fact is, actually, it looks the same as the protein interactions. And it's fairly trivial why that is the case. You can think it through and come to a solution. The question is, to you, will that look more like that? Will it look more like Gaussian? Or will it be something else?
So I would expect the green one on the log-log scale.
Oh! So does everybody understand this answer? There's a power law distribution. Have you heard of that? This is so-called Zipf law. And here is in fact, this log distribution, exactly the one he said. And that, in fact, I assume is the first, the date is missing here, it's the 30s. So that is somewhere This is not the first publication of it. Zipf came up with that much earlier. I forgot to put a CV of Zipf in. He actually studied in Germany, an American who studied in Germany, and that's Zipf's law, 1/x. And the ultimate reality is that any system that evolves in competition, you can do that, you can essentially program a system in the computer, and all you need to put in is this idea of competition. So you grow artificial in the computer, your artificial things that compete for space. And the moment you do that, you get essentially this power law distribution. Okay? So now the question is, are protein-protein interactions competing? And in some sense they do, because there's sort of a limited number of binding partners you can have. So there immediately is competition. Right? So now, in some sense, you get to the next question. The next question is, what is now, what we know today of all the protein interactions that we know is a subset of what exists. And we do know that at this point still the experimental level is at least a factor 10 away from what we believe will be the complete set. If we observe today a Zipf distribution for the number of protein interaction partners, is that a surprise? If we are 10 times away from the truth? So the truth we established here, we expect to be Zipf. But we're 10 times away from the truth. Are we going to observe a Zipf? No. So why not? Whoever says no, why not? Or whoever says yes, why yes? Brings us to the next question. What do you expect a random subset of a Zipf to be? I did not quite understand what you said.
Another exponential and another Zipf distribution. Right?
So the random subset of a Zipf is a Zipf. Does everybody feel that this is true? Yes, it is. So this is a random subset of a Zipf. I mean, yeah, you change the parameters, but you essentially get a log distribution again. So from observing, if we observe today's distribution to be Zipf, then we essentially say by that it's a random subset. Today's distribution actually is not exactly as if you were actually right. We believe this is because the experimental methods have some bias towards picking up certain things, and that is why we specifically pick up not a random subset of a Zipf. Okay? The work, by the way, goes back to something Abner and Yanai Ofran did. Now, Yanai, who was originally the author of the method that predicts the effects of, predicts the residue to be involved in a transient protein-protein interaction, had this idea: when we look at the micro level number of residues with an interaction, we should somehow be able to infer something about the macro level. So, briefly put, proteins that have more interaction sites predicted should have more interaction partners. And this way, we essentially could predict where in the Zipf distribution my protein sits. Okay? Are you happy about that? Is there any sort of problem with that idea? Well, we may make mistakes, and that is not the kind of problem I talk about. We have to try that out. But is there some principal flaw in my argument? Can you think about something? So let's turn it around. There's a principal flaw in my argument. Find it. There's some truth in what I say, but there is something where it would not work. What is that?
Two proteins might bind in different positions. But then on
that is exactly what I'm talking about. So you have different binding sites. So the more binding sites, the more binding partners. That is not where the flaw is. That is where the statement is right.
No, like two A and B could bind on different sites as well.
Oh, that is an interesting one. Yes, that is true, and in fact it's something that I talked about on Tuesday, and that will clearly be a problem, and that could be a major problem. True, but that's not what I meant. Yes.
Because some binding sites, maybe there are more binding partners which are very similar, and they can all bind the same binding site.
Yes, that's the point. So we have one protein, ubiquitin is an example for that. There is a particular ubiquitin that essentially regulates the digestion of proteins. It's the same ubiquitin that binds to 1,000 proteins. It always uses the same binding site and targets these proteins. And that brings us to something, and again I should have a photo here of Marc Vidal. Marc Vidal a while ago introduced this idea of date and party hubs. So hubs again are things that have a lot of interaction partners. Again, whether 60 is a lot or 20 is a lot, this is not well defined. It's just the generic idea. So you have some proteins that have a lot of interaction partners, and some, so the extremes of the Zipf in some sense, right? Now, date hubs are proteins that have the same binding site, and they bind to this partner, to that partner, to that partner. So same binding site, many binding partners. Party hubs is the idea you're standing at a party and you're talking to many people at the same time. So there's somebody there, and somebody behind you, you're talking to all of them. So the party hub is like a transcription factor. Is a protein that at the same time binds to many other proteins with different binding sites. And now we are clear, right? So this means that in principle, what we would expect is that the party hubs have more binding sites. And in fact, this is exactly what we observe as a tendency here. So that's the curve for party hubs, that's the curve for date, and that's the curve for non-hubs. So in principle, okay, these curves are very coarse-grained. There's a lot of noise, prediction mistakes. Proteins can bind in different binding sites. There's a lot of problems, but nevertheless, on our level of the resolution, the pink and the, let me just show you the next one where it is overlaying. So the pink and the gray overlay entirely. So you see pink and gray here, you actually don't, but here you see a little bit of a gray, so that's why you do need the 3D here. They're really falling on the same side, while the party hubs really look different, right? So as a set, and here we used, I believe in the ballpark of 50 hub proteins for party and for date. For a set of 50, we can distinguish them. Whether we can do that on the individual is a different story. Now, where the story got more round is when we predicted disorder in a particular way, so loops, long loops. We saw the inverse. We saw that loops are, in fact, these long loops, disordered loops, you have very often date hubs. So when you have a prediction of the same site binds many different proteins, this is very often actually loopy. This is very often the disordered type of region, okay? So we had one method for capturing the date hubs and one method for capturing the party hubs. And ultimately this is sort of visualized in this graph. Meanwhile, since Janai, in fact, Janai Ofran went on to become a professor by Lyon universities and in fact sort of used the predictions to experimentally verify and find some new hubs. And that succeeded, and a few years ago he published that. That brings us to the end of this part of the lecture.